So thanks for coming on the podcast, Matt.
Of course, yeah. Thanks for having me. Excited to talk about all things de-extinction.
Yeah, so I guess let's start at the beginning then. So who are you and what is Colossal for a lot of our listeners who might not know what you guys are doing?
So my name's Matt James. I'm the Chief Animal Officer at Colossal Biosciences, which might be as confusing as what Colossal is. So we can start with Colossal is this amazing amalgamation of talent and technology. We're a biotechnology firm that started in early 2021 with this thesis of building technological platforms that allow us to restore species from extinction. So everything from historic extinctions to prehistoric extinctions, finding ways that we can bring animals back from extinction in a way that has these meaningful impacts to the ecosystems and to the ecology of the world that support things like global climate change, biodiversity loss, and sort of general conservation. And then as the chief animal officer, I have this amazing privilege of working with all the live animal aspects of what's required in de-extinction, which tends to be things like animal research, reproductive research, surrogacy, as well as animal husbandry for restored species. And then one of my most exciting parts is I also get to do all of the endangered species conservation work, because a really important piece of what we do is apply these technologies that we're developing every single day to the conservation, preservation, and restoration of endangered species.
So quite busy then, in short.
Yeah, very, very busy, but
Yeah.
I like to think, and I think I hear it from some of my colleagues, that I truly have the best job at one of the coolest companies in the world, where I get to kind of interface with every team across the organization, as well as all of our partner conservation organizations. I get to talk to amazing people like you and your audience that kind of share our story. Last year I was doing quite a bit of work on the road to talk about what we do, and it kind of dawned on me that I'm a colossal evangelist, and one of my jobs is to go out there and spread the good word about all the amazing work we're doing.
Because it's on paper, it's almost science fiction, isn't it? It's Jurassic Park in it's probably the simplest way to put it, but it's not far off that, is it really?
Yeah, exactly. I think the two most common reactions I get when I talk to people about Colossal is, number one, is it Jurassic Park? Like, that is, everybody understands that
Yeah.
some of what we do is really kind of rooted in those Jurassic Park type ideas. Now, obviously, we're taking quite a different tact on it. We're not going back to dinosaurs. We're not looking at and also the science is sort of the inverse of the way Jurassic Park worked. But the second one is that scientific skepticism. They go, is this even possible?
Hmm.
And I love that question, because it is, when you pull the curtain back and you get to see what Colossal's working on, it's amazing how possible this is, how close we are, very, very close to sort of the first species being restored from extinction. And it's because Colossal is this amazing intersection of emerging technologies, from genetic engineering like gene editing and precision editing, to reproductive science and artificial intelligence, and animal husbandry, all these amazing sciences that have sort of emerged and are starting to intersect in a way that creates this amazing platform where we can literally reach back in time and bring species back to Earth.
Because certainly in the UK, when I think of cloning, it's like Dolly the sheep. I don't know if you're familiar with Dolly the sheep. That was sort of I mean, this is years and years ago now, and then I've not really kept up with anything since. But I imagine we're a lot more advanced than cloning sheep now.
Yeah, exactly. And so, cloning is one of the key technologies that we utilize in order to successfully de-extinct a species. And that's
Yeah.
what Dolly the sheep went through, is what we call somatic cell nuclear transfer. So that's taking a somatic cell, so like a non-germ cell, or not a sperm or an egg, and pulling the nucleus out of that and putting it into an egg, and literally fertilizing that egg so it divides and becomes an embryo. And you can transfer that back to another sheep, and then Dolly's born. And so that's sort of one of the baseline technologies we have, but we've actually gone beyond that. We've improved it several iterations to improve efficiency and unintended effects. But then we also have a stack of other things that allow us to engineer and edit genomes in a way that we can restore these species that we've lost.
So I was looking on your website, and I think the three main species you were looking at was mammoth, dodo, and thylacine.
Yes.
So why these three? What kind of why do these stand out across all the other animals across time?
Yeah, it's a great question. And when you look at the one of the most terrifying statistics in the world that you'll ever read is, on average right now, we lose about 30,000 species a year to extinction. Now that's across the entire world, from fungi all the way to complex mammals, right? But that's an enormous number. So how do you pick any of them? How is one more valuable than another? That's a really difficult question. But we started with the woolly mammoth as sort of that first project. I think for one primary reason is this technology and this company was co-founded by Dr. George Church at Harvard Medical School. Dr. Church is a world famous geneticist. He's sort of what we call the godfather of synthetic biology. So he's responsible for developing the technologies that make Colossal possible. George had this idea years ago, and he had been sort of working on it as a side project until he met our CEO, Ben, and they built Colossal together. He had this idea of, if we were able to restore a woolly mammoth back to Earth, and we were able to rewild it to the circumpolar north region of the Arctic, then we could actually restore the grassland ecosystem that existed when woolly mammoth still existed. That ecosystem, going back 5,000 years, was as diverse as the African savanna is today. So tons of bird life, other small mammal life, because elephants or mammoths are what we call ecosystem engineers. They spread seeds, right? They disturb soils. They encourage grasslands to grow. So what that would do, in the shortest explanation, is allow us to actually preserve the permafrost. So the permafrost is that permanently frozen piece of soil that exists in the Arctic regions. That permafrost contains about twice as much organic matter or carbon as the rest of the world combined. So as it's thawing due to global warming, it's releasing this organic matter that decays and releases methane and carbon dioxide, which we know are really toxic greenhouse gases. So there's actually these amazing researchers in Siberia at a place called Pleistocene Park that have been working on a similar project to understand, if we were to restore this mammoth steppe grassland ecosystem, what effects could we expect? And they have found that they're able to maintain soil temperatures significantly colder inside the park than outside of their park by restoring these grasslands. So that was sort of the premise of the idea. Is with this is a one way that we could help with carbon sequestration and slowing the warming cycle we're experiencing. Because between today and 2050, we expect there to be as much carbon released from the Arctic as from the United States. So that's, this world's second largest polluter. Is almost doubled up by the Arctic. So finding ways to slow that is a really important piece of that puzzle. And then that was sort of the premise of where the company came from. And we had this wild idea, and people were really excited about it. And we were able to build a seed round of funding and a Series A round of funding and bring all these amazing people to the company. And then we realized, oh my gosh, this platform has such potential. We need to start it to expanding it other places. And we met this amazing researcher at University of Melbourne, Andrew Pask, who has for the last 20 years been working on the thylacine, or the Tasmanian tiger. That is a species that is what we would call an apex predator. So it used to be pretty prevalent very prevalent across Tasmania and mainland Australia, all the way up into Papua New Guinea and New Guinea. And they slowly have been they were receding all the way back to Tasmania. And then they went fully extinct in 1936 following an Australian government bounty program. They essentially were encouraging people to shoot and remove them because there was fear that they were predating livestock. And before they realized how many they had removed, they got down to a number that was so low that the population crashed. And so if you're not familiar with Australian ecosystems, they're just overrun with things like wildlife disease, like Tasmanian devil facial tumor disease, or with invasive species, cane toads, rabbits, foxes, cats. All of these are effects that could be influenced in the right direction by an apex predator present. Apex predators predate on sick and injured species, right? So that helps slow wildlife disease. They predate on those Tasmanian tigers in particular predate on small to medium-sized mammals, which is almost what almost every invasive species in Australia is. So that was sort of the premise for that project, and Dr. Pask had been doing tons of work on that, and so we've sort of injected their lab with a lot of our staff and our funding, and so now we have this amazing collaboration that will allow us to bring the Tasmanian tiger back, hopefully very soon in the next few years. We're hoping. And then finally, the dodo it's the icon of extinction. I think when we think of extinction, dodo is sort of what most people think of. Right? They went extinct in the mid 1600s due to Dutch sailors in introducing invasive species, and people still say, dead as a dodo. If we could find a way to reverse that extinction, I think we can really start to create this optimism in people that we can restore these broken things in the world that most people take for granted as that's done and past. Those species are gone. We find ways to restore them, then we can really start to build a sense of restoration and then responsibility to preserve what's left.
So essentially, it's kind of correcting. I think the theme, I might be wrong in this, but I think the theme with all three of those species is it's largely our fault, or we played a part in their extinction. So it's kind of correcting humanity's mistakes in a way with bringing them back.
Yeah, exactly. Sometimes we say it's trying to amend for the sins of our forefathers and trying to fix what was broken. We're looking at species that could be restored that have this positive effect on our ecosystems and our climate. But we're also driven to extinction by these anthropogenic forces. So human civilization and human development, human hunting played a role in the woolly mammoth extinction. With dodo and thylacine, everybody agrees that was 100% driven by humans.
So the big question is, how do you do it? I don't know if you can reveal exactly how you do it, maybe, but how do we go from talking about this and then one day going to Mauritius and seeing dodos, or Melbourne and seeing thylacines? Like how do we get there?
I mean, it is incredible. It's much simpler than you might expect, but it's also extremely complicated and very a very long process. But it really starts with identifying these species that we want to restore. How can we find genomic information on that animal? So in the case of the woolly mammoth, they did us these huge favors, and they died in a freezer somewhere, right? So their DNA was really well preserved. When we uncover a woolly mammoth in the permafrost, we're able to find some really nice DNA. And also, there's lots of intact woolly mammoth specimens across the world. In fact, we have more than 55 specimens that we've sequenced to create these genomes, and we have more coming. And so that's sort of that first step is, can you create this really nice genome? Can we sequence what a woolly mammoth or thylacine was? And then you sort of have this target. The next step is you identify who's the closest living relative today, who's alive today that would be most closely related to this extinct species. In the case of the mammoth, that's an Asian elephant. For the thylacine or the Tasmanian tiger, it's the entire dasyurid family. So dasyurids are things like quoll and Tasmanian devils, numbats, and then the one that we're using is called a fat-tailed dunner.
Okay.
And then you get to the point of, okay, well, if we call them a genomic donor. So what we would do is we use some of our proprietary genomic analysis software to be able to say, compare these two genomes and identify within the genome of the living species, where do they differ from the extinct species? And then we create gene editing tools to literally go in and snip out those differences and put in our DNA of interest. So in some cases, a woolly mammoth and a Asian elephant are about 99.6% similar. Actually, Asian elephants are more closely related to woolly mammoths than African elephants.
Oh, wow.
A lot of people don't realize that. But however, in the case of a thylacine, they're closest living relatives from that same clade of marsupials, the dasyurids. They're 75 million years removed. So there's a lot more editing needed. So each of these programs has different challenges. We have a really nice high resolution genome for thylacine, but it requires a lot of edits. We have a nice but less HD version of a genome for woolly mammoth, but it requires fewer edits. So that's sort of that challenge. And then once you make the edits to the genome, it becomes similar to the Dolly the sheep example. we're doing somatic cell nuclear transfer or a few other different types of cloning technologies that allow us to take the genes in the cells that we've proliferated from those edits and drop those into an egg, and then let that egg develop into an embryo. That embryo is then transferred to a surrogate. The surrogate would hold that for its gestational term and give birth to the extinct species.
Wow.
A really neat project that we don't talk about all the time, but is really exciting. We're also developing a synthetic solution to that. So in some cases, a living relative might not be able to be the gestational surrogate. So we could create an artificial womb, which is amazing, right? That's where it really very science fiction. And then once they give birth, that's where my team sort of takes over and starts to care for the animal, starts to develop rewilding strategies, creating partnerships with governments, indigenous people, local communities, universities, and find ways that how can we restore habitats that are prepared to receive these extinct species, and then put them back in the wild one day.
So how close to the original are they then? Is it the case that, you know, if you had one of your new ones, so to speak, and the old ones, would they be indistinguishable, or is it a trial and error? Like how close? Oh, do you not know yet?
No, that's a really good question. There are right there are iterations within the cells that we'll need to do so that we can essentially validate that the edits we're making are going to have the phenotypical expression we expect. So some of that iteration occurs in vitro, which is really nice because we can do that quite quickly. But the idea is to get as close to the original as possible. In some cases, right, some people might not realize that your genome or any animal's genome not every base pair in that genome codes for some sort of physical trait, right? Some of them are what we call non-coding regions. So there are specific parts of the genomes that we might not target for editing because they're really there. They play sort of like a buffering role to fight against, you know, unwanted mutations and a number of other things. But we're gonna where the phenotypes that create the animal are expressed, we want to target as many of those as possible. So we will get very, very close to the original. With the way our technology works today, it's impossible to be 100%.
Yeah. That's incredible. You mentioned like contacting kind of local governments and indigenous people, because that must be a pretty strange phone call when you go, well, we want to drop some mammoths in your town, or you know, bring back thylacine or whatever. So like, what's the reaction? Like, have you already approached people for this sort of thing, or not?
With each of the projects that are up and running, we've already plugged into different governmental or community groups, because it's important that we don't get so far down a path and then realize there's nowhere to go with this. So early on in this process, we want to plug into those groups so that we can understand not only is this of interest and importance to you, but how should we go about doing this? What's the most respectful way to your culture and your you know, your sort of norms and values, so that we can be a partner to this? Because our intention is to make these animals so that we have tools to restore biodiversity loss and global climate change. But we need, in order to rewild them and have those intended effects, we need partners. And this is not a, you know, Colossal is not going to save the world by themselves. Colossal is hopefully creating a community and network of people that can together do that.
Yeah, no, for sure. I guess it's really useful not only for the species that you've mentioned, but for species that are on their way to extinction as well. So you've almost got like a backup, if you like, a sort of, you know, save in a video game or something. So it's absolutely bonkers. I think it's brilliant what you guys are doing. Go on. No, sorry, Matt. Go on.
That's what drew me to this company. When I first met Ben Lamb, I sort of said, you know, I think I had the same reaction that everybody has. They go, "Excuse me, you're doing what?" Right? And then you really kind of get into it and you understand Ben's passion and how thoughtful he's really been in creating this company. And then one of the early discussions I had with him, he challenged me to say, find ways to make a difference to the world today. You know, find ways to plug this technology into places where we can really make vast changes immediately, knowing that our technologies take time. And so that's been the most fun part. You know, we've rolled out some amazingly exciting projects in the last year. I think we're really going to be able to make a difference for a lot of different endangered species today.
Yeah. How far can you go back, by the way, as well? Like, so you mentioned mammoths, so that's going to be what, a few Well, quite a few thousand years, but like, is that the limit, or can you go even further
You
back?
you can go rather far. So, right, woolly mammoths, the last one they think went extinct about 4,000 years ago on Wrangel Island off of Alaska in the Aleutian Islands.
Is
But
that it?
really,
4,000 years, really?
yeah, it's not. So think about this. This is something people don't consider a lot. That mammoths roamed the world when the pyramids in Giza were being built. Right.
Wow.
So that provides some historical context because some people think of mammoths like dinosaurs. That's
Hmm.
65 years. We're talking 4,000 years. So it's
Yeah.
really not as far back in history as you think. Now that said, regionally those populations had changed for the tens of thousands of years prior as well. So we have sequences from mammoths as as young as 4,000 years and also going farther back to the 100,000 plus years ago. So the technology reaches rather far back. The limiting factor is the DNA preservation. So
Okay.
mammoths are like I said, that whole idea that they died in a freezer is really helpful because the preservation is really great. If it were a tropical species where there was heat, humidity, sunlight, that degrades the DNA much more quickly. So
Right.
we're unable to go as far back.
Got you. So something like a thylacine in Australia, yeah, you're not going to be able to go nearly as far back.
Exactly. But number one, we don't have to because they were extant so recently. But number
Ah,
two, the story of how we got these thylacine genomes is really incredible. So as the bounty program was underway and thylacines were being poached out of existence by these farmers that were seeking a couple dollars for a pelt, um, they would kill moms that had joeys in the pouch. And so when they would take those, the museums would grab them to create museum specimens. And then they realized, oh, there were joeys in the pouch, and they would preserve the joeys. Now, typically, the typical preservation method at that time was formaldehyde. Well, formaldehyde is really bad for DNA. It actually increases the degradation and makes it harder for us to sequence. But a few of them, for whatever reason, were preserved in ethanol, which is one of the best preservations for DNA. And so we were able to find this one that was sort of labeled the miracle pup. And so we were able to get this beautiful genome from it because it was preserved in ethanol, just probably because that curator ran out of formaldehyde that day. One of those just like really stupid things that made such a huge difference at the end of the day.
little did he know he might be responsible for bringing this animal back because he cocked up not having enough fat. Because there isn't there rumors about them like potentially hanging on. Like, do you subscribe to that, or do you think there might be the odd one, or what do you reckon?
It's funny because we talk about it all the time because thylacine has that cryptozoology idea, right? The
Yeah,
same as
yeah,
here in the States, we have Bigfoot or swamp ape or whatever. Right? If you go to Tasmania, I mean, I just got back last week from a trip to Tasmania. And while we were meeting with community members, we had one of the community members say, I've seen one, like as
really.
recent as a couple of years ago, right? And so it's still, that lore still exists. There is a real hope, which is really cool, I think, for our project that people are so hopeful it still exists
Yeah.
because that means there's such an interest in it. But I would say almost definitively, there are no thylacines left in mainland Australia or Tasmania.
Okay.
There, if there is a remnant thylacine population, it's probably in the highlands of Papua New Guinea. Um,
Okay.
if you think about that, that makes a lot more sense. It's a highly isolated area. Those highlands are almost like islands in the sky. There's the altitude so high, the environment so harsh that people don't tend to live up there. So if there is anything remaining, I would guess it's there, but I'd say it's still 99% that it's extinct. And
Yeah.
if we're in Papua New Guinea, it's a slightly different species than the one that would be found in Tasmania.
Got you. Got you. And the $1 million question, I guess everyone's going to want to know is how soon can we expect to see, mammoths roaming and dodos flapping around?
Yeah, so one of the things that you have to love and hate about working for a guy like Ben Lamb is he has high expectations and he puts it on us to really deliver. So he's really excited to talk about delivering the mammoth as soon as 2028. So that I think that would
Wow.
be incredible, right? And that to me is one of the more exciting parts of it. And when I came on, I was like, yeah, that's what people say, right? You say, we're going to do this really soon. But then you see the speed at which the team here operates and the way that we're able to deliver on milestones. It's amazing. It's really encouraging. With the dodo, we don't have a real target date in mind.
No.
And
Okay.
there's a scientific reason for that. Number one, the way that we described cloning with Dolly the sheep and mammals, sort of pulling the nucleus out of a somatic cell, dropping it into an egg, it doesn't work in birds. You
Oh, got
need
you.
different
Okay.
methods. So we're on the leading edge of what's called primordial germ cell derivation. So literally pulling the precursors of sperm and eggs, culturing those in vitro, editing those, and then injecting those back into a bird that would then mate and create your species of interest. So that is still a technology in the works. But it's what the progress we made since we launched Dodo is really amazing. And for me as a conservationist, if when we crack that nut, the applications to the avian world are going to be
Yeah.
bonkers.
What
The
what was
species,
the sorry, what was there was a pigeon in America, wasn't there? It was wasn't it like the most common bird in the world?
the passenger
The
pigeon.
passenger pigeon, yeah. So presume that's something domestic. I mean, I don't know if people are crying out for passenger pigeons to come back, but they must have been a very important animal because they were ridiculously common, weren't they?
Yeah, the way they used to describe passenger pigeons is that when a flock would fly by, they would block out the sun. There were billions of passenger pigeons.
Yeah,
They
yeah,
were
yeah,
really shot out of existence.
yeah. I don't know if the farmers would thank you for bringing them back, but they must have had a very important role in the ecosystem. So they've got to be important to bring
Yeah.
back. Well, look, before we go, I'll ask one more question, which is I feel like you've kind of already answered this, but there might be other bits you want to elaborate in, which is why should we do this? What you've mentioned about kind of climate change, which is as good a reason as any to bring these back. But yeah, why bring these extinct animals back?
Well, the way that I think about this is that, we sort of talked about our previous generations being responsible for the crises we face today. So we need to find solutions to help reverse those negative effects. But the other piece of it is I think as we create the ability to do so, we also earn the responsibility to act, right? So we are living at this amazing intersection of technology today. It is since that intersection exists, we have the responsibility to act. And Colossal has really, come to the forefront to say, not only will we do this, but we'll raise the money, we'll find the partnerships, we'll do everything. So we're very driven to find those solutions. And one of the ones that doesn't get as much attention as I think it should is the biodiversity crisis we face today. Like I said, there's a very famous ecologist that estimates we've lost about 30,000 species every single year. Now, when we're talking about the high order mammals and birds, we're looking at numbers in the hundreds, but still that's an unacceptable rate.
Yeah, yeah.
Uh, if you think of an ecosystem like a Jenga puzzle, it's stable because there's all these different pieces that create the foundation in the center and the top. As we continue to drive species to extinction, we're pulling these blocks out of that Jenga puzzle, and that ecosystem is becoming wobbly and losing stability. There will be a point where we pull on a block and the whole thing collapses. So what we need to do today is find ways to reverse the biodiversity crisis, not just slow it down, not just stem it. That's an important role that traditional conservation organizations have been working on and need to continue to work on. And we hope our technologies will support. But we need to find ways to put blocks back into the Jenga puzzle to really help create that foundational stability so we don't have a massive catastrophic collapse of ecosystems, because that will affect humans more directly. That's food chain security, right? That's water, that's climate, all of those things that we need to worry about just to survive in this world. So if you don't care about the intrinsic value of nature or animals, that's fine. If you want to be self-centered, care, because it will come to bite you in the ass one day.
Yeah, no, I think you're completely right. We're a reactionary race when we should be far more precautionary. Like we just think, oh well, this thing's cocked up. Let's try and fix this. When yeah, we are heading for a major crisis if we don't try and avert it. So look, Matt, absolutely fantastic to talk to you. I can't wait to kind of turn on the news one day and see mammoths roaming around. Where would they be released? Where would you put them back?
But we're working with a number of different governments. We
Yeah.
plugged in I think Alaska is a great opportunity. We
Yeah.
are an American-based company, so
Okay.
American Arctic would be a target. But
Great.
Canada, Greenland, Siberia all have different Arctic regions that could be candidates for that.
Awesome. I went to Alaska last year for a filming job, so mammoths would have made that far more interesting as it already was. But yeah, it's been an absolute pleasure. So thanks for coming on.
No, thanks for having us. And anytime that I can evangelize, just call me.
Will do. Cheers.
Thank you.